Welcome to the lesson on DNA transcription and translation. In this lesson, we'll be discussing how DNA is decoded to make proteins through the processes called transcription and translation. Let's go ahead and get started. Transcription and translation means or equals decoding DNA to make proteins. Let's start with transcription. During transcription, we make a temporary copy of one gene. To do this, we have the chemical called messenger RNA. You've heard of RNA in a previous lesson. Ribonucleic acid as opposed to DNA, which is deoxyribonucleic acid. So RNA, ribonucleic acid, but specific kind here called messenger RNA. And the abbreviation for this is mRNA. And this is the code to make a protein. mRNA is only a single helix. And in RNA, the base thymine is replaced by the base uracil. So it uses uracil instead of thymine. And so instead of a T, which pairs up with A, you would have a U that pairs up with an A. Let's take this DNA strand, and you have the coding side of the DNA strand here on the top, and this is just a section of the DNA strand, which would be a gene. And what's going to happen here is that the mRNA strand is going to code with the coding side of the DNA strand. So what would the letters be for this? Well, what would pair up with T would be A. What would pair up with A is not T in RNA, it would be U. And so on down through. So we should get these letters paired up with the TAC-AT-GACC-ACT coded side. The mRNA for that gene would be AUG, UAC, UGG, and UGA. Note that I did those in series of 3 bases each. More to come on that. And then from there on, the mRNA goes through a process called translation. And translation is decoding the RNA to make a protein. And then to do this, the mRNA leaves the nucleus and travels to the ribosome. All genes in all organisms begin with a start code AUG. So note that the start code of our mRNA strand is AUG. We'll talk about stop codes here in a little bit. Okay, next page here. So we're starting with our strand of mRNA, which again, AUG, UAC, UGG, and UGA. And what we do is we separate it into those sets of 3, and those sets of 3 are called codons. So the codons for our mRNA strand would be AUG, our second codon would be UAC, our third codon would be UGG, and our fourth codon would be UGA. The next thing we do is we determine the anticodons, and this is called transfer RNA. In order to determine the anticodons, use the codon and circular table that you'll find on page 311 of your text to determine the proteins. Okay? Now, I'll take you through this real quick. The anticodons, let's back up a second here. The anticodons, or transfer RNA, the tRNA, you come up with the opposite letters for each one. So what codes with an A would be a U. What codes with a U would be an A, and what codes with a G would be a C. And you do this for each of the 4 codons in this particular example. U would be A would be U, and C would be G. UA, GC, GC, and UA, GC, AU. To figure out the proteins that are produced, you take your codon and you use this circular table, and I'll show you how to do that here now. You use the codon itself, not the anticodon. So you take a look, our first codon here is AUG. So you start out in the middle, there's our A, then you work your way to the outside. U, and then G. And you see the three letters that start this protein are MET. And this, of course, the name of the protein itself is methionine, but they can't fit all that in there, so they just use abbreviations. And note the word that's associated with right here. It's called start. This is the start protein. Okay? So again, those first three letters will always be AUG. So your start protein here will be MET. Go to codon 2, UAC. There's U, and then A, and then C. And our first three letters there are TYR, and so the protein produced would be tyrosine. For codon 3, UGG, start with a U, G, and G, and our first three letters there are TRY. And that's tryptophan, which you might, by the way, recognize. This is the protein that makes you sleepy after you eat turkey. Codon 4 is UGA. So U, G, and then A. And notice that this is a stop code for the gene. So every gene has a start code, AUG, and then it's going to have one of these stop codes that are intermixed throughout here, which you can see, I believe, are right here, and then there's two right here next to each other as well. Okay? All right, so that's how it's done. I know this is confusing, but let's take a look at a video by Hank, which will explain this perhaps even more, and also do a good job with some animation on this as well.